If you think pattern hair loss is only a hormone problem, you may be overlooking what happens inside the cells that keep a hair follicle alive and cycling.
I thought androgenetic alopecia was basically about androgens shrinking sensitive follicles until the hairs become thinner and shorter.
That is an important part of the picture. But this review asks what may be happening inside those follicle cells, especially inside their mitochondria.
The structures we usually call the cell's powerhouses?
Yes, but that nickname is too narrow. Mitochondria produce energy, regulate reactive oxygen species, handle calcium, influence differentiation, and participate in programmed cell death.
So if mitochondria become dysfunctional, the follicle could have trouble with more than just energy supply.
Exactly. A hair follicle is a dynamic mini organ. Its cells must proliferate, differentiate, survive, communicate, and move through repeated phases of growth, regression, and rest.
And androgenetic alopecia may involve disturbances in that whole cellular system.
That is the theme of Progress on Mitochondria and Hair Follicle Development in Androgenetic Alopecia, Relationships and Therapeutic Perspectives.
So, the paper asks whether mitochondria help connect familiar hormonal signals with the cellular changes that determine whether a follicle keeps producing healthy hair.
Right. Mitochondria do not replace the androgen story. They may be one place where energy, oxidative stress, signaling, and cell survival converge. To see why that matters, start with the hair cycle. Follicles move through anagen, the active growth phase, catagen, the regression phase, and telogen, the resting phase.
So, keeping hair is partly about keeping the right cells doing the right job at the right point in that cycle.
Exactly. Hair follicle stem cells contribute to regeneration. Dermal papilla cells provide growth-related signals, and keratinocytes help form and support the follicle.
Where do mitochondria enter that process?
At several levels. They supply ATP, help manage calcium, influence apoptosis, and generate reactive oxygen species, or ROS.
ROS sounds like damage. Wouldn't less always be better?
That is one of the key corrections in this paper. Excessive ROS can contribute to oxidative damage, but mitochondrial ROS can also function as normal signaling molecules.
So the same category of molecule can be useful at one level and harmful at another.
Yes. The review describes mitochondrial links to major developmental pathways, including WNT and beta-catenin signaling and Notch signaling. Those pathways help regulate follicle development and cell differentiation.
Then trying to eliminate every bit of oxidative activity could disrupt normal biology.
Precisely. Hair follicle cells need sufficient energy and controlled signaling without tipping into damaging oxidative stress or inappropriate cell death.
And that makes mitochondria more like a control center than a simple battery.
That is a useful way to think about the problem. This paper is a review, so the authors did not recruit one new group of patients and test a single treatment against a control group.
Then what did they actually do to answer the question?
They brought together prior research on mitochondrial function, hair follicle development, hair cycle signaling, mitochondrial disorders, androgenetic alopecia, and possible treatment strategies.
So instead of one experiment, they are building a map from many earlier studies.
Exactly. They examined how mitochondrial energy production, ROS, calcium regulation, apoptosis, and mitochondrial proteins connect with hair follicle biology.
And they compare that map with what is known about androgenetic alopecia?
Yes, one important connection is evidence that dihydrotestosterone, or DHT, can induce mitochondrial dysfunction and oxidative stress in dermal papilla cells associated with androgenetic alopecia.
That gives a possible bridge between the hormone side and the mitochondrial side of the story.
Correct. The review also considers experimental models in which mitochondrial genes or proteins are disrupted, because those models show what happens when mitochondrial signaling or metabolism no longer works normally.
So, abnormal follicle development after a mitochondrial disruption can point researchers toward mechanisms worth investigating.
Yes, but the evidence comes from different systems, including cell studies and animal models. The review supports a mechanistic framework without proving that every pathway has the same role in human androgenetic alopecia.
So, the method is strongest for connecting pieces of biology and identifying plausible targets, not for declaring one new therapy clinically proven.
Exactly. The resulting picture is that mitochondrial dysfunction can interfere with hair follicle health through several routes at once.
Let's start with the most obvious one: energy.
Hair follicle cells need ATP for proliferation and differentiation. When mitochondrial function is impaired, reduced energy availability can make normal growth and regeneration harder to maintain.
Then there's oxidative stress.
Yes, excess ROS can damage cellular components and promote apoptotic signaling. The review connects this with DHT-related mitochondrial stress in dermal papilla cells.
But the paper also says some ROS are necessary, right?
Exactly. In mice lacking mitochondrial transcription factor A in keratinocytes, mitochondrial ROS production is disrupted, and important Wnt and Notch signaling is impaired. Hair follicle development and differentiation are affected.
That is very different from oxidants are bad, antioxidants are good.
It is. The useful model is a regulated range, not a race toward zero.
What else changes when mitochondria malfunction?
The review discusses mitochondrial proteins involved in follicle development and cycling, including CRIF1, AIF, and MPZL3. It also describes metabolic changes as hair follicle stem cells differentiate.
So the follicle's energy system changes with the cell's job.
Yes, mitochondria participate in energy production, signaling, differentiation, and apoptosis. Disturbing those functions can be associated with abnormal follicle cycling, weaker hair structures, or impaired regeneration in different disease models.
That makes the mitochondrion less like one cause and more like a hub where multiple problems can meet.
Exactly. That systems-level view is the review's strongest contribution. That systems view also changes how the treatment section should be read. The goal is not simply to boost mitochondria or swallow an antioxidant and expect hair to return.
So what kinds of approaches does the review discuss?
It covers strategies that interact with mitochondrial biology, including minoxidil-related effects on calcium and ATP synthase activity, low-intensity laser approaches, mitochondrial antioxidants, targeted drug delivery, microneedles, regenerative approaches, and mitochondrial transfer.
Mitochondrial transfer sounds far beyond ordinary hair loss treatment.
It is presented as an emerging concept, not an established treatment for androgenetic alopecia. The idea comes from research showing functional mitochondria can be transferred to damaged cells in other disease contexts.
And what about antioxidants, such as MitoQ or N-acetylcysteine?
The review treats mitochondrial antioxidants as promising directions and notes research on N-acetylcysteine in early-onset androgenetic alopecia. But it also warns against trying to remove ROS completely.
Because ROS are also part of normal follicle signaling.
Exactly. That gives us a practical decision rule from this paper. Do not translate oxidative stress matters into the less ROS, the better.
And don't forget, mitochondria matter.
Right. The more useful perspective is that androgenetic alopecia involves a follicle whose hormones, signaling pathways, metabolism, oxidative balance, and cell survival machinery interact.
So, future therapies may become more precise by targeting specific cellular dysfunction instead of treating mitochondrial activity as one simple dial.
Exactly. The strongest insight is that healthy follicle function appears to depend on mitochondrial balance, enough energy, regulated signaling, controlled oxidative activity, and appropriate cell survival at the right stage of the hair cycle.